So firstly, the agenda for today. So this is the very quick welcome, me giving you a bit of an update of what to expect. As I said, my name is Lauren Carson. I'm Head of Research Development for UK Biobank. And then following this section, we will be going through the data overview, going through the details, value and highlights by our wonderful epidemiologist, Megan Conroy. And then following on with a section about use and access, talking about how to use the RAP and some useful tooling by our product manager who's focused on imaging, Dr. Ollie Gray. And then I'll wrap up the session today and talk a little bit about researcher funding, as well as credit programs we have available for researchers, followed by a brief Q&A section. If you do have any questions that you would like to ask any of the presenters or myself, feel free to do so by going over to the Q&A section next to the chat section, so that we'll be able to answer your questions potentially on camera. If not, we do have a team answering some questions in the background as well. So moving over to my colleague, Megan.
Hi there, I'm Megan Conroy and I'm an epidemiologist at UK Biobank, and as Lauren says, I'm going to give an overview of the data we've released this week. So as you can see here, this just shows which categories all the data we've released have come in. So quite a broad range of data that's gone out. And I just want to highlight before I start that for all of our data that we release, we do put together quite in-depth resources which are available on Showcase. And so I really recommend if you're interested in the more technical aspects of the data, that you go onto Data Showcase and look at the resources there. And we also have details on the community forum as well, so it's worth popping over to the community forum. So I'm going to start with our headline data, the biggest data release for this data, which is the sleep questionnaire. So this questionnaire was put together in response to researcher requests for more detailed sleep data. And we worked with research groups to put this together. This data release is available for 179,000 participants. Now, the questionnaire is actually still open to participants, so that number will rise slightly over the next year or so as more responses come in, but it won't be many. But just to say, this isn't the final number. And as far as we're aware, this is the most in-depth sleep survey of its kind to be undertaken. So the group that put this together, they used validated questionnaires to collect data on sleep patterns and disorders, work and sleep, sleep conditions, restless leg syndrome, sleep consequences, and associated lifestyles and behaviours. So the reason that this data is so important was the sleep data we currently have is not very comprehensive. Health records, the NHS-linked health records we have do not capture sleep disorders well. We have 5 questions of recruitment that cover snoring, insomnia, chronotype, sleep duration and daytime sleepiness. And we have some accelerometer-derived variables around sleep duration and chronotype. But as you can see, it's not the best for phenotyping sleep disorders. So this questionnaire really captures a lot of information on insomnia, restless leg syndrome, obstructive sleep apnea, sleep-wake phase disorder, shift work sleep disorder, narcolepsy, parasomnias, and REM sleep behaviour disorders. So this questionnaire has really been put together to help researchers to characterise heterogeneity within the disorders and reduce misclassification. And then when you combine this data with the wealth of data that UK Biobank already has, the genetics, the imaging, proteomics, metabolomics, environmental data, it will really allow researchers to investigate the underlying biology of sleep disorders. And then with the linked health data, look at the health consequences of that. And with such a large data set, a sample size also will have enough for variation in normal sleep as well. So yes, it's very exciting data, and we're really looking forward to seeing how researchers use it. And also our participants are also very excited to see how you'll use it as well. We have very involved participants, and here's one quote from a participant just showing how much they're looking forward to seeing the results coming out from this data. So moving on, we are updating our imaging data. So we now have 80,000 scans from the imaging visit available on Showcase. We're getting very close to that 100,000 that we aim to scan. The FIMRIB brain IDPs, this is a pipeline that generates the researcher-ready phenotypes from the brain images, has been updated to 64,000. And we also have new connectome data. This has been returned to us by a research group where they've generated connectome data for the brain images. And just for those who do use the brain images, we have restructured the brain image showcase page category. So it's easier to identify the provenance of each data. So we have one category for UK Biobank provided images, one for the FIMRIB provided IDPs, and another for the connectomes. So we hope that that restructuring will help you find things on Showcase. The next imaging update is for our carotid ultrasounds. So up till now, you've needed to purchase a proprietary software to be able to use the ultrasound videos. And because of that, they've not been well used in research. So we've converted our videos to the .mat format. So it's non-proprietary, you no longer need proprietary software, so far more accessible. And these data will come with a three-dimensional matrix, which represents two-dimensional image through time, number of frames in the video, the metadata associated with the video, as well as coordinate data for plotting. So yes, we're really excited to see what people do with this data now that it's far more accessible. We've also updated our genetic data. So for the whole genome sequencing Dragon pipeline, we have PLINK and BGen format now available, which has been requested for a while. So we really hope that that will open up this data to people to use. And we also have machine learning corrected PVCF. So this the research group who generated the whole genome sequencing have done this to more accurately call the regions of the whole genome sequence that were the quality wasn't as good. So there's more information on how they've done that on Showcase and on the community forum. The next updated genetic data is on the polygenic risk scores. So the PRSs have been returned to us by Genomics PLC, and they have updated to version 2. There's two variations in the polygenic risk score. We have the standard polygenic risk score, which is generated using models that haven't used UK Biobank data within it. So the standard polygenic risk scores are available for the whole cohort. And then the enhanced polygenic risk scores, the PRS data, the data used to generate the PRSs did have some UK Biobank participants in it. So it's only available for the subset of UK Biobank that was not included in that testing data set. We have 3 new traits: resting heart rate, total triglycerides, and total cholesterol added to the standard PRS. And they've updated the testing subgroup and therefore also the enhanced subgroup, with people being added and taken away from both of them. So they're not directly comparable to the previous version, the people that the enhanced PRSs are available for. And there's been updates to the Crohn's disease, ulcerative colitis, and prostate cancer polygenic risk score. And Genomics PLC have put together a GitHub, which is really detailed around how they've generated these PRS, with the link available is on Showcase. Another research return is VO2 max from the exercise ECGs that were undertaken in a subset of participants at recruitment. So these researchers have used a multi-level framework which incorporated the 3 phases of the ECG to estimate VO2 max. So this is the first derived phenotype we have from the ECG data. So hopefully it will really open up that data to people who are interested in cardiovascular fitness, but haven't had the expertise to be able to use the raw ECGs before now. We've had some sample results. We have a sample application currently ongoing to assay integrated human herpes virus 6 within the cohort. So human herpes virus 6 can integrate within the genome during a primary infection, which then leads to vertical transmission rather than just standard infectious transmission. HHV6 usually integrates into the telomere region of the chromosome and can result in sudden shortening of the telomere, which is hypothesized to be associated with adverse health outcomes. So this is the first set of data that we will get back from this sample application. And they've provided us with 5 options for the whole cohort around if they likely have integrated HHV6, direct, repeat, and unique only, which are two measurements that they undertake, unlikely to be and confirm negative. So that data is available now, with more data to follow probably early next year, I'd imagine. And we've also put together invitation data. So this is data that researchers have been quite interested in. And so it's data on who we've invited, when they've been invited, and the acceptance or non-acceptance of the invite. So our enhancements, we invite via email. I think it's about 370,000 of our participants have provided us with an email address. So for example, our questionnaire invites all go out via email. So this data reflects when that email was sent and when they accepted via the email. It should be noted, though, that participants don't need an invitation to undertake the questionnaires. They are made available in the participant portal. And so they may log in and undertake the questionnaire without an invitation. So you may notice that there's a slight discrepancy between who we said we've invited and who's actually undertaken the questionnaire. So this will allow you to really look into non-response bias with that questionnaire data. It's not yet available for the imaging assessment. We are working on it, but we've used a different invitation protocol for the imaging assessment. So it's a bit more complicated to put that variable together, but we hope to have that released in the near future too. We're always making sure our data is as accurate as it can be. And we have both our data analysts are always checking the quality of the data, and also we have feedback from researchers around data quality as well. So we have some corrections and amendments to the data. So the first of all is we have restricted the OMOP data set. This is because there were some quality issues around the mapping from the codes that we have to OMOP, so it's not accurate. There's a full details have been made available in the news section on the community. If you do still want this data, you can still have it. You just need to be aware of the data quality issues. So you just need to get in contact with us so we can make it available to you. If you are interested in OMOP or other common data models, we do actually have a survey live at the moment on the community forum, which would be great if you could fill in. So, because we're trying to understand how much interest there is in common data models and whether they'd be interested in us generating these common data models. So please do go and answer that question, the survey, if this is something that you're interested in. There were some errors with dates of assessment, which have been imaging assessment, which have been corrected. And we had some address history inaccuracies that our data analysts identified, which have been corrected. So if you have used the address history data, we really strongly advise you to go and download the new variables to use in your analysis, as they are more accurate. Other updates is we've updated the mortality data. So we now have about 55,000 deaths in the cohort. So is 10% of UK Biobank have now sadly passed away. We've updated the location at assessment. We've standardized our geocoding methods, and the location data for the imaging assessment has been added, as well as the census area information, which was only available for baseline previous to this, is now available for each follow-up assessment. And we've also updated the fluid intelligence score. So that is the updates to the data. If you have any questions, as Lauren said, please pop them into the Q&A and we will answer them as and when we can. And I'm just going to pass over to Dr. Ollie Glary now to talk about how to access the data and tooling for the data.
Okay, great. Thanks, Megan. I'm Ollie Glary. I'm a product manager at UK Biobank, and I'm going to be talking about how to access the data that we've released through the UKB-RAP. You'll recognize this workflow if you've kind of been on UKB-RAP before. So we've released the data, and to access that data, you're going to need to have a UKB-RAP project. If you created a project recently, then you should be able to refresh your project to include the most recent data. There's a link going up in the chat to with a guide on how to do this. In that project, you can collaborate with the specified collaborators on your AMS project. You can explore the data, and you can perform downstream analyses. You can also analyze bulk data files, such as images, and create tools that you can use in the future. So to explore that data, you may wish to use the cohort browser. There's a link going up in the chat with further instructions on how to do that as well. You can filter here for cohorts that have specific features and extract those cohorts to a file for using later on to put into an applet or a Jupyter notebook. Variance browser is also available, and this is based on the genotyping array sequencing. And this will allow you to filter based on participants that have specific SNPs that you're interested in, or even participants without those SNPs. With respect to performing kind of downstream analyses, processing bulk data, creating tools, you'll need to be familiar with the DX toolkit. We've named and classified some functions here that should be useful to you in doing that. If you're interacting with the UKB-RAP interactively through applets or other tools, you'll need these functions to do things like manage your session, so logging in and out of the RAP, to managing your data, so uploading and downloading data from your project space to your compute space, your instance space, and also navigating your project files, and then finding and managing, terminating jobs using the manage and run functions there. Applets are tools that enable effective parallelization of code and repeatability of analyses to be applied to the data. These are particularly useful when you have a list of, say, file inputs and a standard set of code to apply to those files. This could be an automated image segmentation pipeline, for example. To build applets, you'll probably find the DX app wizard useful. This essentially requires you to specify some inputs to the applet, for example, some files, some data files, some outputs from the applet, so files that you're expecting to be chucked out the other end, and any package dependencies you have, and the specified bit of code that you actually want to run. We provided a really simple example applet on the UK Biobank GitHub account, and there's a link to that going up, and that should be a fairly useful starting point for you. And yeah, there's a link that link should be in the chat for you to click on. Okay, also on the UK Biobank GitHub account, we've provided notebooks. These describe exploring and exporting the data through Jupyter notebooks in Python and R and through RStudio. And they also provide some kind of simple analysis kind of demonstrations there as well. These are great for kind of developing a more fundamental understanding of the structure of the data and the different methods that one can use for accessing and exploring. You can follow that account and you can star the repository. There's a link going in the chat to that to be notified of new releases of additional content in the future. To help make access to the sleep questionnaire data that UK Biobank's released in this refresh of the data, we provided a notebook that you can use to export the sleep questionnaire data to your project space. This is written in Python and is available within the Jupyter Notebook Python folder of the Access repository that I just mentioned and linked. With respect to the imaging data that we've released, we've recently published an article on the UK Biobank community. This details the different types of images and provides useful links to other resources, including more detailed descriptions of those image types and the acquisition protocols and processing protocols that have gone into generating the data. And they also include descriptions of how to start instances that are preloaded with brain imaging tools like FSL and FreeSurfer. We're also on the brink of releasing an article describing how to use the 3D slicer tool with UKB-RAP. This tool enables image segmentation, model training and testing for automated segmentation, and allows visualization and rendering of images for your publications and things like that. This work builds on a seminar that was provided by DNA Nexus in December on working with the Mono open source framework in UKB-RAP. So feel free to kind of go and have a check out of that as well. Lastly, we have also just published a guide on running a MATLAB user interface on UKB-RAP. This guide takes you through from starting MATLAB, installing toolboxes, and saving your configuration for future instances using Docker snapshots. So hopefully that's of use to you. I've noted some resources here on the UK Biobank community and on the UK Biobank GitHub account, but there's also some useful other resources from DNA Nexus, both for their training videos, their documentation on their Gitbook, and also their more general documentation that should help you find information on how to utilize UKB-RAP for your specific use case. And posting forum updates, forum questions on the UK Biobank community is a great way to interact with other researchers and to get help from UK Biobank data analysts and other people like that. So I'm looking forward really to hearing and reading all about the exciting research that comes out of the data that Megan's provided, and I hope UKB-RAP can help you do that. Just like to thank quickly the funders and of course the participants that are at UK Biobank. And now I'm going to hand back to Lauren to talk about funding and the support that we can provide.
Hi, so I hope you've thoroughly enjoyed both the presentations by Megan and Ollie. I certainly have. So I'm going to very briefly talk to you a little bit about funding and researcher support that's available. I'd like to remind everyone as well, if you do have any questions for either of the other speakers or myself, feel free to put them into the Q&A section, which is very close to you, but not within the chat function. So if you could do that, that would be fantastic, because it'd be great to have that conversation with you. So firstly, I'd like to talk to you about fee structure. You can see here the fee structure that we have. And this is for all projects that start at UK Biobank are for 3 years in duration, which can be extended later. We have Tier 1, Tier 2 and Tier 3 data. Tier 1 data is charged at £3,000 for those 3 years of access, and extended pro rata, as with all of the tiers, if you do extend your project. And this contains all of our core data sets. So this is things like our health outcome phenotypes, links to health data, web-based questionnaires, etc. Tier 2 data contains everything that was within Tier 1 data, however, also includes biochemical and hematological assays, as well as other enhancements that we have, as well as the imputed genotypes, for example. And that's charged at £6,000 for 3 years of access. And then finally, we have our Tier 3 data set, which includes everything that's in Tier 1 and 2, as well as our imaging data set, our other large-scale assays, our whole exome sequencing data, our whole genome sequencing data, etc. And that's charged at £9,000 for the 3 years of access. We also would like to remind you that you are more than welcome to collaborate with other institutions as well on your project. It doesn't have to be based solely out of one institute, and it's wonderful to see so much international collaboration that goes on in our research projects. This does come with a slight administration charge for each institute that you add at £1,000 per institute for that 3 years. And then if you do extend, there is again another admin charge there. And then finally, we are always encouraging applications from researchers who are based at institutes in lower income countries, or are students based anywhere in the world. These projects are these projects are offered at a substantially reduced rate to encourage researchers from these regions and of these stages in their career at £500 for the first 3 years of access, and then can be extended. There are certain extra stipulations with regards to the student applications. Further information about this can be found on the community. So, as I've mentioned before, there are some ways that we can support you financially with regards to your projects. So we have 2 wonderful credit programs. We have the platform credit program, which offers early career researchers, so that's a researcher within the first 4 years of their academic appointment or as a student, or any researcher based at an institute in a low or low to middle income country, £1,000 credit to support and cover any costs associated with using the UKB-RAP. So this is your compute and storage costs, and this comes courtesy of Amazon Web Services. After you've spent that initial 1,000, if you do require further funding, you can then go on to apply for enhanced funding. Very similar to the platform credits program sponsored by AWS, we have the UKB-RAP transition credits program. Now, we know that the move to the RAP for some researchers has been financially challenging, and this credit program is to support you in your transfer over to the UKB-RAP. And this is courtesy of Welcome. This is open to all researchers who have an active ongoing project, and you can, again, as I said, apply for £1,000 worth of credit, very similar to the platform credit program, to cover costs associated with compute and storage on the UKB-RAP. Both the platform credits program and the transition credits program will, if you are eligible and approved for these credits, your credits will be added to a wallet within the UKB-RAP environment. And further information about both of these can be found on our website. And then finally, we have the Global Researcher Access Fund. This fund is to cover the application costs of that £500 fee that I mentioned in the previous slide for researchers based at institutes in low to middle income countries. A full list of these countries that are eligible, again, can be found on our website. This fund is, again, to increase the access for researchers who may historically not have been able to access data sets such as UK Biobank due to financial constraint. And this has been very kindly donated to us via some of our pharmaceutical companies who work with UK Biobank extensively, including AstraZeneca, J&J, etc. We would also like to draw your attention to the fact that our community page has now had a facelift, and my wonderful colleague has just put up a little box in the top corner there. You can connect through to that by clicking on the link. And this, our wonderful researcher community has a wide range of guides and tutorials within there on how to support yourself with your work on our access management system, which is a way that you deal with the admin side of your project, as well as the UKB-RAP. There's also easier links in there now to browse the data showcase. We also have some fantastic new articles in there associated with working with our studio, working with Jupyter Notebooks, the bulk data and tooling libraries associated with that, as well as some great getting started guides for PIs, including lots of additional information around billing and billing queries that you have with regards to using the UKB-RAP. In here as well, there is a fantastic forum. This forum allows you to collaborate with your peers, provide peer support, answer questions other researchers may have, so that we can really build this fantastic community of researchers using UKB. And last but by no all means least, you can also submit requests within there to UK Biobank if you do have queries specifically associated with your project itself, which comes through to the wonderful access team at UK Biobank as well. So please do go check it out. It's really had a major facelift and is got some really new exciting information in there as well. Finally, I'd like to draw your attention to our next webinar, full details for which will be coming soon. This is associated with the sleep data that Megan wonderfully presented earlier. And goes through some of the key findings since the release. This has some guest speakers in from the University of Oxford, both Simon and Hang, as well as Joe, who's our wonderful health data group lead from UK Biobank. So now we're going to move over to the Q&A section. So I believe the first question, apologies, I'm just inviting my other speakers back on stage. So the first question, apologies. So the first question we're going to go to is from Rick Henderson. And I believe, Ollie, this is going to be a question for you. How many of the 80K participants with neuroimaging have repeated imaging, so such as the second or third time point?
Yeah, so thanks, Rick. So we have data available at the moment from the first repeat of the full imaging protocol for 5,000 participants. This was the COVID participants plus some extras. So those collected during 2021 to 2020 to 2021. We're expecting the first 20,000, another 20,000 addition to that at some point in 2026. It'll probably be halfway through 2026 around then. And we'll give you lots of interesting, exciting news about that when it's finally released. Our future timelines page on the website will give details about when that's being released and what's being released as well.
Fantastic, thank you, Ollie. The next question we've had through is, how do you decide what new data to add? Is it entirely from researcher feedback, or are there other factors?
Do you want me to take that one as well, Megan, or do you want to? I'll start with that one. So it tends to be covering the data sets that cover a large proportion of the cohort, so they're preferable, and data that is kind of going to be widely used by the rest of the community. So we're always interested in kind of furthering the engagement of the whole researcher cohort in the data that we release. So those two things are kind of real high priorities for us.
Fantastic, thank you, Ollie. The next question that we have through is, how can researchers get in touch to suggest and work with UKB to develop questionnaires? So I'm happy to answer that one, actually. Drop us an email. Let us know that you're interested, because that's the best way for us to then start that dialogue with you to understand if you do want to come and work with us. We can then pass you on through to Joe Holliday, who I mentioned before, and we can try and work out the best way of incorporating your questionnaire into the next questionnaires that go out if it's appropriate. So I hope that answers your question. The next one is from Shaman. Is the 500K metabolomics data included in this release? Megan, would you like to take that one?
No, it's not included in this release. I believe it's in the next, either the next release or the release after. But yes, you will know about it when it's coming, because we will be sure to advertise that.
Yes, we will make a very big noise about that. It is a fantastic data set. Unfortunately, couldn't quite make it into this cut. So I don't believe there are any further questions to be asked, unless anyone has got very quick fingers in the audience and are able to get a question in. So I'm just going to hold for 2 seconds. There's one that's just snuck in. Is there any follow-up data available for cardiovascular disease, such as myocardial infarction, such as imaging, plasma, blood markers, etc.? Also, several SNPs were not part of the list in the UKB website. Is there any update on inclusion of more SNPs?
I can certainly take the first part, maybe, or a bit on the imaging, and maybe Megan could take the bit on the health data, if that's all right. Split it out. So with respect to imaging, there's no kind of participants haven't been specifically imaged, whether where they have had infarctions or any kind of radiological or clinical signs. There is a chance that people have had those and cross-tabs with the health linkage data that we have could provide you insights about which participants. And like I said, the cohort browser and filtering and specifically identifying people that have specific features that you're interested in could help you with that. Megan, about updating?
Yeah, so I suppose for health data at the moment, it's the hospital inpatient data, which is limited to diagnosis only. We are always looking at other data sets that we can link to try and get more information. It's not a quick process. It takes a long time to sort everything out and get everything linked. But if you do have suggestions of data sets where this information may be available for, we're all ears. Please drop us an email. Particularly around the plasma and blood biomarkers, that would be great. About the SNPs, we have no more plans to do any more genotyping. So what's on available now, directly measured, I think there's about 850,000, and then imputed to millions. But obviously we now have whole genome sequencing, so you should be able to pick up every genetic marker within the whole genome sequencing.
Excellent, thank you both. The minute I said about not being able to see questions, lots of them popped up, so apologies to those in the audience. The next question is there HCP/cluster support available while to use while analysing?
Yeah, so the DXCore toolkit that I talked about, it provides kind of HPC kind of comparable compute, so you can access large clusters of individual instances. It does work differently from a kind of traditional HPC that you would get at an institute, and kind of designing your code and workflows so that it kind of fits better within a parallelized environment rather than kind of a long serial analysis pipeline will make things cheaper for you and make kind of workflows more easy. Having kind of breakpoints at which you can stop and have like resilience against failure of the whole pipeline is also going to help to make you your pipeline a lot more kind of cost efficient and effective.
Great, thanks, Ollie. The next question, will methylation data soon be available?
I think that there is an ongoing project to do long read sequencing that's been announced fairly recently, which is very exciting. I don't know about soon. I think it will be planned out and put in the future timelines. I don't actually think that the project is I think it's in early stages at the moment. So.
So the project that Ollie is alluding to, there was actually a press release a couple of months ago. It's with Oxford Nanopore Technologies, who are undertaking a research project with us alongside Decode in Iceland, and further information can be found about that on their press release on their website. But as Ollie said, these projects take time to run, ingest the data, and then deliver it out to you at a really high quality. So we will, as Ollie said, put it on the future timelines pages, and we'll get it to you as soon as we can. There is another question, just 1 second, with regards to what is the estimated the ETA on the returns access system. You were told early 2025. Thank you, Jay, for that question. I'll actually take that, seeing as access is my department. We apologise that there has been a slight delay with this. We are still trying to hone exactly how we are going to undertake this process. Please bear with us. As soon as we have the returns process sorted out with regards to the UKB-RAP, we will let you know, and we will let researchers know who've contacted us about this as soon as possible. So thank you. And then there's a lovely other question with regards to the postdoc trainees. Are they eligible for student discounts? Again, another question for me, sorry. Unfortunately, no. So what we, for the student applications, fees, so that £500 for 3 years of access, that is only for master's student and PhD student level. Unfortunately, it's not eligible for postdocs. The However, the credit program sponsored by AWS, Platform Credit Program, is available for postdocs who, as long as you're within the first 4 days of your first academic appointment. So I'm sorry about that, but hopefully there's a little bit that we can do there for you with regards to your compute and storage costs. So we're now going to bring this webinar to a close. We thank you so much for your attention, and please do, if we haven't had a chance to get to your question during this session today, we will try and get back to you after the event as well. So thank you so much for your time joining us today, and we hope you have a lovely day. Take care.
Thanks.
Bye.